All right, it is my pleasure to introduce our distinguished Grand Round speaker, Dr. Gareth J. Morgan. Dr. Morgan is a professor of medicine at the NYU Grossman School of Medicine and director of myeloma research at NYU Langone Health. Dr. Morgan holds an impressive array of degrees, including from the Royal College of Physicians and Pathologists in London, for medicine, hematology, and leukemia. His research focus has been on characterizing the myeloma genome to identify disease drivers and predisposition loci. His team has most recently focused on characterizing tumor cell interactions in the bone marrow niche, contributing to disease progression from monoclonal gammopathy of undetermined significance to myeloma. Dr. Morgan has identified a number of genetic prognostic variables and developed integrated clinical and molecular staging systems for myeloma. He was the first to identify the association of ApoBec, or apolipoprotein B mRNA editing enzyme catalytic polypeptide-like mutations with MAF overexpressing subtypes of myeloma. More recently, he has been following up on initial work characterizing the importance of MYC translocation by investigating the impact of structural variants on myeloma pathogenesis. Dr. Morgan is a prolific physician researcher and has authored well over 500 articles in high-impact peer-reviewed journals. It is an honor to welcome Dr. Morgan for today's Medical Grand Rounds.
Thank you, Michael. I have to say the honor is all mine. It's a delight to be asked to talk to you guys who I've got a lot of respect for, and hope I can do the request the honor it deserves. So I've tried to make it a talk that's kind of generally relevant and makes a point about the pathogenesis of cancer in general and heme malignancies specifically. So I think we should aim to cure multiple myeloma. It's getting a bit old to hear that it's an uncurable disease that has a relapsing and remitting history and you die within 2.5 years. It was never a great paradigm for approaching the treatment of a cancer, and I think it's even less appropriate now. And I think in order for us to achieve a cure and to make progress, we need to understand the genetics of disease, initiation, progression, and use this information to optimize treatment. And just to make it clear to people, 30 years ago, really there was no information about the genetics of myeloma, other than there were 20 myeloma cell lines, which people did cytogenetic studies on. And from that point, we've moved on quite substantially. I'm going to make the point that there are many new treatments that utilize the immune system. And if we're to use these treatments appropriately, we need to fully understand the impact of the disease on the immune system. And achieving MRD negativity is an essential prerequisite for cure. So response-adapted therapy using sensitive tests to monitor the disease makes sense. Further, once you've achieved MRD negativity, I think we should use biological knowledge to design therapies that manipulate the evolutionary history and the biological features of the remission state. And so nothing makes much sense in absence of a consideration of evolution and the concept of humans developing in the kind of exposure to constant challenges from the environment in terms of infection and having to develop an adaptive immune system that makes the most appropriate kind of response to the environment. And so really, this is my simplistic view of the immune system, where really you're modifying your germline sequences to enhance your immune response. But if you change your DNA sequence, there are inherent dangers in that to the generation of mutations and generation of mutations within the immune cells, which can generate cancer. So while there's a system to protect against those variants, even a low frequency event as you get older will potentially have some impact on the proportion of the population. So immunoglobulin gene rearrangement, class switch recombination, and somatic hypermutation are all intrinsically dangerous processes for the organism. And this is a good cartoon of myeloma. We understand that there are two broad types, one characterized by rearrangements generated by class switch recombination to either NSD2, cyclin dependent kinases, or the MAF genes. The other 50% of cases are characterized by hyperdiploidy. These are generated in the lymph nodes. The plasma cells migrate to the bone marrow, where they evolve, form focal lesions, then extramedullary disease, and in end-stage disease can grow in the bloodstream as plasma cell leukemia. So these were the first linear models of myeloma development, showing these immunoglobulin rearrangements and then the path or the passage through monoclonal gammopathy of uncertain significance through smoldering myeloma to myeloma and extramedullary disease. And clearly genetic features drive the disease. To the right in this diagram, copy number, mutations, DNA abnormalities are important in that. And at one level, applying what you can think of as a pigeonhole principle to define when variants occur is very important. So we know that translocations are present in 100% of the tumor cells, suggesting they are really etiologic factors. But then these other variants, the deletion 13, deletion 17p, 1q gain, these all occur at increasing frequencies as the disease progresses. And these increasing frequencies are used to imply that they're driving the disease forward. And the more abnormalities you have, the later in the disease process you are. But we wanted to kind of develop on the ideas of cytogenetic abnormalities and just those limited variables seen from classic metaphase cytogenetics to carry out a sort of genome-wide approach to try and use DNA and RNA molecular profiling to improve our understanding of myeloma and the disease process. And so the first papers really defined myeloma as a disease of the RAS-MAP kinase pathway with 20% of either K or NRAS, so about 50% of myelomas have mutations in the RAS pathway. But in addition, you have BRAF, and down here there's a couple of MAP kinase active mutations. But really, it's about the major drivers are RAS, and these are the positions of the mutations. And so the mutations that are seen occur in the appropriate hotspots of mutations seen in other cancers, arguing they truly are driver mutations. There's a different spectrum of mutations in the different etiological subtypes, and it's tried to show that here with the differences between the 11-14s, 4-14s. I guess people were sort of underwhelmed by this knowledge, as while we can show that you can target BRAF, you switch it off and people will go into a remission, they all seem to relapse quite quickly. And rather than the mutational spectrum, I think what we learned from the initial studies was more around the fact that not all of the mutations are present in all of the cancer cells, and they tend to occur at a variable frequency between 90% down to 25 to 30%. And again, this suggests that there is intraclonal heterogeneity, and this could act to drive evolution, selection, and cancer progression, in contrast to what we thought happened before, where there was a strict linear progression in mutation analysis. So it's very analogous to what Darwin proposed for the evolution of species. And rather now we have a model for myeloma development, again based on the transition from Muggus to myeloma to plasma cell leukemia, but based on these branching evolutionary pathways and the formation of distinct ecosystems, so that when you're treating the disease, you need to think of it as this collection of different cells, each of which have different biological properties. It would be important not just to invoke this from studying the bone marrow. And so we, with some German colleagues, spent some time biopsying tumor lesions, because myeloma is nothing if not leukemia with focal lesions distributed throughout the bone marrow. And so we were able to do sequencing of paired sites from the same patient. And you look at the two sites and you can see the genetic characteristics of what might be at the chest wall are different to what you might find at the iliac crest. Again, consistent with this evolutionary process and diversification that's driven by position as well as focally at any one site in the body. We did more of this, trying to pair the lesions from presentation, not only with other lesions at presentation, but to what was present at relapse. And the guys did a great job of this and were able to show that many relapse samples are more likely to have lesions shared with the focal lesions than with the general bone marrow, suggesting that there's increased evolution and diversification at the sites of these focal lesions of disease, which then disseminate to the rest of the marrow. And on the right here, you can see what are the mechanisms leading to relapse, where you can have expansion of a single cell repopulating the whole bone marrow, or you can have two major subclones at different sites which have reconstituted the marrow, or the third model is where there's spatial dominance and regional evolution, so that you have these models of single cell expansions, clonal sweeps, diversification, hidden and dormant cells, all of which undergo clonal competition and really lead to the formation of these complex systems that exist in relapse refractory disease. And the factors that govern the progression through this involve the mutation rate within the stem cells, the size of the population at risk, and the fitness of the variant that is generated. So most of the time, cells are at an evolutionary disadvantage, and it's only the ones that give advantage that are actively selected for and grow out. You would then ask the question, okay, so what is happening at relapse? And our initial studies showed that structural variants and inactivation of tumor suppressor genes by biallelic deletion was a common mechanism. More recently, as part of the same collaboration, this very large series of whole genome sequencing was done on paired, newly diagnosed and relapsed samples. And I guess in the past you would have been anticipating finding interesting new mutations, but actually it's somewhat underwhelming, the information you get from this, which were specifically done in IMiD-resistant cases. But largely, there are very few novel mutations. Most of the mutations that were found were those that were seen at presentation, potentially at a slightly increased frequency, but little in the way that was novel, with only 1 or 2 genes that were specifically mutated in the pomalidomide, lenalidomide refractory cases. Again, pointing to the importance of other mutational processes than single nucleotide variants. Okay, so to end this section of the talk, why is this relevant? So diversity ensures the evolutionary fitness of the cancer cells. So it acts as a complex system, where if you perturb one part of it, the other will grow out. The diversity increases with time and with exposure to treatment, and it certainly supports adaptation to drug resistance. So long-term low-dose exposure is a good way of generating drug resistance. It also says if you take a sample at, say the shoulder compared to the pelvis, that you can have sampling bias. You may get different mutations at one side to the other. You will also expect differential responses at sites based on these differences in biology. The idea of clonal tiding is important. If you were exposed to one treatment at one time with linear evolution, you would expect to be resistant to that treatment forever. But with clonal tiding, if you've been off the treatment for a while, it's possible that a sensitive clone could come to dominate the marrow, and you might be sensitive to something you've been exposed to before. But importantly, when it comes to targeted or mutationally targeted treatment, at some level, it's doomed to failure, because if you put a pressure onto something that's present in only a proportion of the cells, the other proportion of the cells will inevitably grow out and lead to relapse. So maybe targeting biology is better than targeting mutation. All of this Darwinian ideology is kind of largely based on a constant mutational rate over time. And so if you think of what happens in the clinic, there are various ways that you can relapse, sometimes explosively with rapid progression of aggressive disease, and on other occasions, just slow increase in the level of the same clone. And so actually you need to think of other molecular mechanisms and what could explain these cataclysmic changes in behavior. And the way to describe that, rather than this stepwise evolution, is punctuated evolution. And these would tend to be due to catastrophic events that deregulate multiple genes simultaneously. So you have to think of what kind of molecular mechanisms may do this. And this is from Evan Rusted and Francesco Mauro, the work with Ola there in Miami, describing these different types of structural variants. That one's called chromothripsis, where a region gets blown up and then reassembled randomly, where chains of lesions are formed with deletion of the regions in between, and templated insertions where you gain copy number at the sites of the chains. And these have important implications for deregulation of cancer drivers. So this you would expect to activate tumor suppressor genes, this to activate oncogenes, and this to be very penetrant, because like these kind of mashed up regions have the potential to deregulate many genes at a single site, and so confer prognostic impacts. And again, this is a sort of description of those lesions. Here's the copy number losses. Here are the multiple gains of copy number in short regions, and these are the deletions. So these, not surprisingly, associate with regions of active transcription, and these do not. But interestingly, if you look at the impact of chromathripsis on prognosis, it occurs in some 20% of samples. So it's a lot of occasions it can happen, and it's quite penetrant in its effect. And it could prove to be a very useful prognostic factor, as it is. And you can see the adverse prognosis in terms of PFS and OS in really quite a large series of cases. And it's an independent prognostic factor that I think if we could find it regularly in the clinic would improve our capacity to predict outcome. So again, what is the clinical relevance of these molecular events? And so we have a spectrum of events that we understand, and maybe we can use them for disease classification. And so again, this is Ola's paper looking at the early stages and progression of MGUS, the pre-malignant phase of the disease. And you can see that frequently you see increases in the M spike over time, but just using disease bulk and behavior to define a disease doesn't make sense. So maybe we can use myeloma defining events. And so if you look in smoldering myeloma or MUGUS and you find RAS mutations, FAN46C mutations, translocations to MYC or APOBEC signatures, these are reflective of these cases that are in evolution. And so if you look early, you might be able to predict these cases that are going to pursue an inexorably progressive course. And again, you can use this information. And I kind of think Francesco and Benedict Oben from Belgium did this publication suggesting that really there's only two conditions. There is a stable myeloma precursor condition you might call MGUS, and the other cases with the myeloma defining events are all likely to progress in the fullness of time. And while you might not want to intervene early in this second case, all of these may benefit from earlier intervention. And there's a series of clinical trials investigating this hypothesis currently. The other thing to say is that myeloma falls in the middle of the spectrum of mutations in cancer. It's not simple like pediatric leukemia or CML, and it's not complex like sarcomas, lung, etc. And so it's a good model to understand these more complex malignancies. And if you look at its kind of mutational signatures, it's shaped by 8 mutagraphs, and it's worth pointing out this APOBEC mutational signature, which is associated with the DNA modifying events that are important in class switch recombination and somatic hypermutation, suggesting it may be important in the context of that. And there are two clock-like mutations that we'll come to, or mutational signatures that we'll come to in a minute. Clearly, if you acquire APOBEC early in the disease, and this is a project done by Eileen Boyle, if you develop an APOBEC signature during the smoldering myeloma phase of disease, you're more likely to progress. Again, suggesting it's an important thing that we need to understand more about in terms of defining disease entities. And to kind of bring us to that more, this really was Francesco's work using clock-like signatures which occur at a constant rate over time, and then using the rate that you see in the tumor samples in front of you, you can extrapolate back to when the tumor was initiated. And the math of it is far too complex for me. But the implications for biology and epidemiology are really important, because the implication is that for the majority of cases, the initiating event occurred between 20 to 30 years before the clinical presentation, suggesting that the tumors have been in the marrow evolving for a very long time. And if you want to understand the early phases of disease, you have to work out a way of studying those directly by extrapolating what's happening at that disease time point. And so, again, using this pigeonhole principle and using copy number acquisition as a way of defining or timing when a lesion occurs, you can have a pre-gain where you have 2 copies, and then post-gain where you add additional copies. And so you can time when mutations are occurring in the context of this. And rather cleverly, Francesco kind of looked at the acquisition of these aid APOBEC signatures over time, and was able to define an evolutionary trajectory towards multiple myeloma based on the relative frequencies and timing of the aid and APOBEC process. And so there seem to be differences between African Americans and European Americans in terms of the frequency of these events. So jointly between NYU, MSK, CC, and the University of Miami, we have this SMART study where we're aiming to resequence several thousand cases of myeloma, impute their racial origin, and then to look at signatures to understand how the signatures develop and be able to look at what's happening in the early stages of disease to understand why African Americans have a 3-fold excess of disease compared to Europeans. The other thing you can do is to use molecular variants to define prognosis. And if you use the classic features, this was a shock to me because we'd sequenced like 1,200 cases, and we had full details on nearly 800 cases. But when you look to see what was penetrant, it turns out that there's very little in the way of single nucleotide variants that are penetrant. And really what you're seeing is the impact of biallelic p53 inactivation and gain of chromosome 1, with most everything else not standing up to multivariate analysis. And so this is this situation for 17p minus and p53 mutations, which is located on 17p. If you have either just loss of one copy of either allele, either mutation or loss, you don't have a negative impact on prognosis. But if you lose the front and the back breaks, you're in some kind of problem. And so importantly in the clinic, using molecular approaches to defining what's going on rather than simply IFISH makes every bit of sense. The same with gain of chromosome 1. Here is chromosome 1. You can lose copies on the P arm, but the major prognostic factor is gain of a region around here on the long arm. And if you gain one copy or two copies and look at the impact of that on prognosis, you can see that really most of the adverse outcome comes from gain of two copies. And the question's always been, what are the genes on chromosome 1q that are important? And so we tried to map the sites of deletion and gain. And you find there are multiple foci of gains, and they're labeled up here as A1 to or A2 to 9. And here you can see that most of the time, though, you gain everything, but in a proportion of cases, you have focal gains. And at some level, the suggestion is that the molecular mechanisms lead into these two types of events are different, because this is where most of the poor prognosis lies and not in this group. But again, trying to define the genes. So you have these multiple sets of experiments have given these multiple different driver genes, but the hypothesis has to be that it's the overexpression of a set of genes from chromosome 1 that are important, rather than there being any one specific driver. And we're kind of ongoing working on this currently. Oops. So this is a sort of unifying molecular approach to cancer progression in multiple myeloma, showing each of the chromosomes, the gain of the odd-numbered chromosomes, the abnormal class switch translocations early in the disease, the hyperdiploidy, the role of the germinal center in driving immortalization of some cancer initiating cell, which then goes to the bone marrow and is driven to evolve by a variety of different mutational events, including many of the ones we've seen. Initially, there's a diffuse growth pattern. It then develops focal growth. These are important in determining relapse, where translocations and structural events are also very important in mediating disease progression. And so, like, as a molecular geneticist, you'd be very happy with that as a classification, but it ignores one thing, and that is cellular biology and the fact that myeloma orchestrates the development of an abnormal bone marrow microenvironment where there are tumor promoting cells, tumor suppressing cells. There is a stromal niche and an osteoid niche, all of which are modified by the cancer cells over time. So it's a back and forth process driven by genetics in the bone marrow impacting changes in the microenvironment. And so when you think of the evolution of these things, so mutation rate, size of the population, fitness, stemness, but there are two important features that really need to be in any system, which is the selective pressure of the immune system in immune editing, and then when they're on treatment, the selective pressure of the treatment. And so we have 4 kind of major classes of drugs, including the IMiD drugs, which are immunoacting, the proteasome inhibitors, which are also immunoacting in a way, antibodies that can either be kind of directed against an epitope or be T-cell fixing antibodies or CAR-Ts, all of which suggests that in addition to genetic prognostic factors, there may be important immune prognostic factors, and we know little about that. But using our current treatment, we need to get away from this historical paradigm of relapsing, remitting, uncurable, refractory disease, death, to something that's more presentation, frontline therapy, remission, maintenance of remission for as long as possible, and death from a different cause, incomplete remission. And arguably, this is a feasible and potentially possible approach. And it's where Dr. Landgren's obsession with MRD and MRD monitoring comes into being. So this is the, you start treatment, you have the iceberg, you enter a complete remission, but when you're in a complete remission, you have variable amounts of disease. And we know the deeper response you get, the better you do long term. And so this is a very useful clinical paradigm that we should all be adapting now, with response adapted therapy being a critical way forward. But what I'm hoping to just make this point is that it's not just the MRD monitoring that's responsible. We need to understand the biology of minimal residual disease states so as we can manipulate them. And so think of this as treatment, the bottleneck. And what you're trying to do for people in remission is manipulate the cellular content to generate a better prognosis and keep people in remission for a longer time. And you're effectively manipulating competition within the bone marrow niche. And so with a slide that Faith made originally, using the tumoricidal impact of your drug early on is important, but over time you want to use immunomodulation to control the disease. And I'm going to make the case that we're in that setting already and say how we've tried to understand it. So in an original experiment, we wanted to take cases at the different stages of myeloma and look in an unbiased fashion at the disease and their microenvironment. So the first attempt was to use whole bone marrow RNA with gene expression profiling across all of it. But because we had tumor cell and whole bone marrow, to take a signature of these cells, extract it from the whole bone marrow, and then use a computer algorithm to generate each of the cellular components. And the program we used was CyberSort, which I can tell you does not work on the bone marrow. And there was quite a lot of modification and new cell types we had to put into the imputation. But the bottom line was one population stood out as having an adverse prognosis, suggesting that there is a high-risk microenvironment, and there were some typical features of this high-risk microenvironment, which were an increase in monocytes. It was kind of unsatisfactory because I don't think CyberSort really is fit for purpose, and I think there are better ways, more accurate ways of gaining more important insights. So the second attempt at the similar analysis was to use CYTOF, and again, a not insubstantial analysis, not up to the many hundreds of cases that we did in the first analysis, but certainly substantial enough to look. And you could define different clusters of the bone marrow, and we were able to look at these over time and at relapse. And the bottom line was you see changes in T cells with them going down, and macrophages and monocytes increased in number, again pointing to the important role of the myeloid lineage as well as the lymphoid lineage. Okay, so that's as way as introduction. So to get some data, the addition of daratumumab to Velcade, rituximab, and Dex, the standard upfront regimen, has dramatically improved outcomes, but patients can still be resistant to these quadruplet regimens. This is Ola's single-arm Manhattan study that used daratumumab, carfilzomib, Revlimid, and dexamethasone. And you can see following these cases over time that you start to get many people into this green component where there's no detectable residual disease. You need at least 5 to 6 cycles to maximize the number of responses. But again, for once, we're getting to deep, deep responses in the majority of patients. And the question is, what governs response to these cases? Because some people stay MRD negative long-term and others relapse quite early. And so to kind of, well, I'll come to the experimental details in a second, but you need to know how Darzalex daratumumab anti-CD38 works. And basically it fixes complement macrophages and NK cells to the surface of the myeloma cell, leading to their death. But it also has this immunomodulatory effect where it affects Tregs, Bregs, and MDSCs. And so using whole bone marrow samples from a substantial number of people that remain either MRD positive or relapse or become MRD negative and sustained MRD negative, which we predict to be long-term good performers, we were able to compare the two groups using single-cell RNA-seq. And basically, we use Surat to do this. We quality controlled and we have good quality data. You integrate all of the data so you can interpret it in its entirety, and then you do the analysis. And you start to be able to demonstrate the bone marrow and the cellular content of the bone marrow in terms of these PCA or Tisney plots with each of these individual populations within the bone marrow clearly visible. And so you can generate a reference population and you can compare at presentation and after treatment the content of the bone marrow, and we had sufficient samples to split it into good prognosis and poor prognosis at both time points. And this is T1 and T2, so during at presentation before you've been treated and after you've maximized out your treatment. And what stands out in this is the presence of significant differences in monocyte populations. You see changes in the T cell populations, but at kind of minimal levels, making them kind of somewhat more difficult to be accurate around. And there are substantial changes in the B cell populations as well. But I think it was the monocyte populations that stood out, certainly in the context of the signals we'd had previously. And so you can split monocytes into classical monocytes that have phagocytic capacity. There's an intermediate stage, and then the non-classical ones, which are inflammatory. And here you can see the differences that T1 time point between the good and the bad responders. And here at the second time point. And so these, I think, are fascinating, certainly given the mode of action of daratumumab and how this kind of may work in that setting. But if you take the monocyte population and then repeat a PCA, you start to separate out different populations. So you can see distinct differences between the good and the poor responders, not only in the frequency of CD14 and CD16 monocytes, but look, in this population, very distinct missing population at that site. And here, nothing in that position, whereas here you have a substantial population. So the treatment and the response is substantially mirrored in these features, suggesting that they may be important in mediating the different outcomes. And so this is a better summary of the data, where in the good prognosis MRD sustained, you have these IL-1 beta high, GAD45 high, CD14 monocytes that can phagocytose the myeloma clone when exposed to the daratumumab, that then revert to a non-classical phenotype with more inflammation when you look at the second time point after they've responded, where the 14 monocytes that are CTLA-4 high are present both at diagnosis and after 8 cycles in the cases that remain MRD positive. And so it's always important then to go to the literature and check what other people find. And there's a very nice paper from a European group showing that if you do look specifically at monocytes, you can see changes analogous to what we see during the development of the disease through the Mugger smoldering myeloma, myeloma transition. So I'm going to finish there and kind of try to summarize. So once myeloma is initiated, it evolves by Darwinian features which can inform and optimize the use of our therapy. Geneticists think that the genetic features of the cancer cell drive the evolution, but it's clear that you should take a genetic and biological approach with the two interacting to generate disease progression. But it's clear that the modified microenvironment does impact outcome. Monocytes are key features of this high-risk microenvironment. We can achieve deep responses with current treatment, but we need to enhance these by better, more kind of informed use of our immunotherapy approaches. And manipulating monocytes offers one approach that could be useful in the clinic. And so I'm going to thank the team here at NYU Langone, where I used to work at UAMS, Brian Walker at CRAB, really say a thank you to the guys at Miami who've been great collaborators, including all of the guys here, and for Ola and Francesco, who are kind of old friends, and the people in UK and Germany. Okay, I'll stop there and say thank you.
Well, Dr. Morgan, I want to thank you on behalf of the department for presenting such a clear grand rounds, and what a thought-provoking discussion. If you could turn your slides off for a second so we can see each other. I'd like to maybe start by asking the first question. I think you have to hit stop sharing. There you go, perfect. So so many different things that you said were so intriguing, but the concept of a clock, a molecular clock that starts maybe 20 or 30 years even before all of this begins. In light of, I think, 2 weeks ago in Nature, there was a discussion of mutational clocks. Have we identified the clock itself? that, or are we close to understanding what that is that sort of starts the ball rolling?
So in myeloma, it's very clear that the thing that sets the ball rolling is the acquisition of a structural variant to the IgH loci and switching on one of 3 different oncogenes. There may be a kind of initiation set of biology, but we don't understand that. So really, I should say that those structural lines set the clock ticking. There's a specific clock-like signature, 2 clock-like signatures in myeloma that really do seem to occur at a standard rate that you can extrapolate back to when the disease starts. All that stuff from last week was kind of interesting and important, and I think set the ball rolling for understanding signatures in many different types of cancer that hadn't been possible before. But we've been able to do this in myeloma for a while.
Thank you. At this point, I'll open it up to anyone who would like to ask any questions. Dr. Seckers, would you like to comment, or Dr. Langren?
Sure. So Gareth, outstanding talk. I mean, it's just absolutely comprehensive, and I love how you take this almost teleologic view of evolution of cancer over time. I wonder if, I'm fascinated by the evolution from MGUS to frank myeloma. Do you have any idea what is the greatest pressure on cells that causes them to go down what I refer to as the Grimrose path towards frank cancer?
So it's very difficult to answer that. I kind of really didn't think so much about immunoediting in the past, but with the use of immunotherapy in the clinic, I think it forces you to consider what role the immune system does around removing the weak variants, and most acquired genetic variants make the cells weaker. And so it just becomes this inevitability about you need to access the niche, and anything that allows you to exist in the niche better than the normal plasma cell is going to be selected for. So you're constantly in sort of competition between the normal and malignant cells. And eventually the malignant cells can exist in the niche better. And when they can do that, they then start to displace normal cells. And then inevitably you get a mutation that increases the rate of mutation, because I think what's obvious is that you can never have enough mutations with the standard mutation rate to ever get cancer. So you have to have some form of genomic instability. And in myeloma, the only thing we have are these APOBEC mutations. And like, I'm obsessed currently with the idea of retrotransposons and the role played by APOBECs in suppressing retrotransposons, and whether there's any form of input from retrotransposons in actually facilitating the expansion of these clones. There's no answer to that, but it's fascinating to speculate on.
Gareth, maybe I may ask a question. First of all, congratulations to a beautiful talk, excellent presentation, and thank you so much for citing all the work from our group as well. I was going to ask you 2 questions, one more of a clinical question and one with a hypothesis concept, and one based on data. So could you share your perspectives on the data that has been cranked out more recently, the past 6 months or so, on the role of these highly effective therapies that are used, how they impact the microenvironment, and the potential risk for second malignancies, and the role of CHIP? A lot of people are interested in that. And also the second question is, you share slides, different perspectives on the literature. You had this longitudinal trajectory with monoclonal gammopathy and smoldering myeloma and multiple myeloma, but you also shared data on the role of APOBEC activation, chromatopsis, structural variants, and the split into 2 different entities. So the question is there, is the history of smoldering myeloma about to end? Are we talking about precursor disease or myeloma early on? Will the intervention start earlier? Will the disease be initiated therapy based on genomics? What is your thinking? Those are the two questions.
So I know you and I share thoughts on the kind of early phases of the disease, and it's good to collaborate with you on these things. And I actually think there is no such thing as smoldering myeloma or kind of aggressive phase chronic myeloid leukemia. I mean, in our case, you have MUGUS, which I think is stable and genomically bland, whereas in contrast, there are myeloma defining events which you can use to avoid using a 10% bone marrow cutoff as some kind of biological truth, which makes no sense to me whatsoever. I think what I was trying to make the case was that you would never do a therapeutic intervention in something like MUGUS. But if you were sort of myeloma in evolution, where you have all of the features present, you treat early, you anticipate doing better long term. And so I think as a field, we have to move towards really tightening up those definitions so that we could actually do a population-based intervention of some sort.
Thank you very much. At this point, I'm going to ask everyone to direct their attention to the chat, to click the CME tracker so that you can get credit for today's grand rounds. Again, I want to thank our distinguished visitor, Dr. Morgan, for an excellent grand rounds and everyone's participation. I wish everyone good health, and we'll see you all next week, if not before.